Okay, hello everyone. Let's begin with today's session on. The NPTEL course name is Enzyme Sciences and Technology, and this course is taken by Professor Vishal Trivedi from IIT Guwahati, and I am the NPTEL TA, Chaitra Todankar from IIT Bombay. So, in the last lecture, we learnt about introduction to enzyme, its classification and nomenclature. Today, we'll learn more about or the protein structure, and and how do we analyze these different protein structures. Okay, so enzyme or protein is made up of is enzyme are mostly protein in nature, and these proteins have different stages of structure, like primary protein structure, secondary, tertiary, and quaternary protein structures. So in the primary protein structures we have amino acids, and these amino acids are joined together in a peptide bond. In secondary structures we have complex structures like alpha helix and beta pleated sheets, and these two secondary structures further form the tertiary protein structures, which are more complex and 3D form of structures, and multiple tertiary structures or subunit together come and form quaternary protein structures, as shown here. So the basic unit of the protein are amino acids, and this is the general structure of amino acids. So we have a amino group here, as you can see. To the chiral carbon, we have a carboxyl group here. So this structure has both amino as well as carboxyl group. That is why the name amino acids. And we have a central carbon, which is a chiral carbon, and to this chiral carbon we have four different moieties attached, one of which is amino, and the third group, which is the R side chain. So different amino acids comprises of different R side chains. So this R moiety here changes with different amino acids. So this is the general structure of amino acid. The average molecular weight of amino acid is taken to be as 110 Dalton, and accordingly we can calculate the molecular weight of protein depending upon the number of amino acid present in the protein. Now what are the different R groups which are present in the amino acid? So we have non-polar R groups, we have polar R groups, and we have electrically charged R groups. So in the non-polar R groups are mostly aliphatic in nature. So we have the simplest amino acid here is glycine, in which the R group is hydrogen, whereas there are other amino acid which has different aliphatic groups. So in non-polar we have aliphatic R groups, attached. So this is alanine with simple methyl group. Then we have valine with CH3 group attached. Similarly, we have different aliphatic amino acid as seen here. Then we have methionine, which has a sulfur group attached in between two methyl groups. And we have here few aromatic amino acids which are uncharged or non-polar. The two aromatic amino acids which are uncharged or non-polar are tryptophan and phenylalanine. And proline is one of the cyclic amino acid which doesn't have a chiral carbon atom here. Okay, and in polar amino acids we have serine, which has a alcohol group, OH group. Similarly, threonine also has a hydroxyl group here. And then we have a cysteine. Cysteine is very unique and very important amino acid. It has a sulfahydryl group. It has a SH group. And in polar amino acid, we have a aromatic amino acid, which is tyrosine, which is the structure is similar to phenylalanine, but we have a hydroxyl group attached here. Similarly, other polar amino acid are asparagine and glutamine. Then we have electrically charged amino acids which could be either acidic or basic. That is, it could be either negatively charged amino acid or positively charged amino acid. So in the negatively charged amino acid we have aspartic acid and glutamic acid, whereas in positively charged amino acid we have longest side chain residue, which is a lysine residue here with the amino group. So this is a positively or basic amino acid. Then we have arginine here, which is also a basic amino acid with a guanidinium group here. And then we have histidine. Histidine is also very important amino acid, and it is found in most of the active sites in the proteins. So these are the different amino acids based upon R groups. Next, if we want to analyze different types of amino acid, we could use different techniques like thin layer chromatography. This is a very basic and very important technique. Thin layer chromatography here so this is this rectangular sheet could be either a thin layer made up of silica gel, or it could be the stationary phase or the stationary layer here. It could either be silica gel or it could be filter paper, Whatman filter paper. So in thin layer chromatography we have a thin layer of silica gel mobilized on the slide or any aluminium thin film, and that's why the name thin layer chromatography. So on this rectangular sheet we then spot different amino acids, and then they are separated based upon the distance they travel. So the basic procedure is the first we run the solvent front. So this is the distance which is run by the solvent front, and this solvent front could be any solvent in which your amino acids are soluble, and they will run a distance. Let's say they will run the distance ds, and these are different amino acid which are spotted here, and they will also run some distance d1, d2 and d3 as shown here. So the overall distance can be calculated by using this formula of Rf, where Rf is the retention factor, and this retention factor is distance traveled by analyte or different amino acids which are spotted upon distance traveled by solvent front. So different amino acids will have this different Rf value depending upon the R side chain. So charged molecules will tend to move faster as compared to uncharged molecules, and uncharged molecules will stay here. And now this will depend on which solvent we are using. So everything should be taken into account, and we can actually separate different amino acids. So we can run a standard amino acid solution here, and then we can match this different amino acid with the standard solution, and thus we can analyze that in our unknown sample what all amino acids are present. These amino acids in the primary structure are joined together by peptide bonds. So this is the this is one amino acid here, and this is the second amino acid. So a peptide bond is formed, which is CONH bond. A amide bond is formed with a hydrolysis of water, and this peptide bond it is the core of the primary structure of primary structure of proteins. And this peptide bond angle, it has the property of this peptide bond angle has a property that it can rotate. Okay, so the NC angle and CN angle can rotate. So this is the peptide bond in the amino acids, and then we have rotation here. C alpha C angle and NC angle can rotate, and these angles are known as phi and psi angles. So C alpha C angle is known as psi, whereas N and C alpha angle is known as phi. Okay, and these two angles are in the primary structure of protein. These two angles are considered these are the dihedral angles which are considered during the Ramachandran plot analysis. So as you can see here, this is the Ramachandran plot, and on the y-axis we have psi degrees, whereas on the x-axis here we have phi degrees. And different secondary structures like alpha parallel beta sheets, alpha anti-parallel beta sheets, alpha helixes will have different dihedral angles, and these dihedral angles, as you can see here, these are the different values of the dihedral angles. So for alpha helix we have phi angles to be minus 57, whereas the psi angle here is minus 47 degree. Okay, so alpha helix will fall here. Okay, in the Ramachandran plot they will fall here, and the blue shaded region, this blue shaded region is nothing but the permissible angle. These are the permissible dihedral angles which are allowed. So if we have a X protein, and if we have a X protein and it is and that peptide, small peptide, is following a left-handed alpha helix, they should fall in these angles. Okay, then only they are accepted as alpha helix. Similarly, we have different dihedral angles, phi and psi angles for anti-parallel beta sheets and parallel beta sheets. So this is the basis of Ramachandran plot, and it is a very important tool for analysis of different types of unknown proteins, or in order to validate your model experimentally as well as through computational models. Next we wanted to next we wanted to see how the protein are sequenced. So there are different stages for protein sequence analysis. So the first step in protein sequence analysis is so we have a globular protein, as you can see here. This is a globular protein. First we need to break down. So the first step is the breakdown of this protein. Okay, so and this breakdown is done by breaking disulfide bonds. Disulfide bonds. Okay, and this can be done by using two reagents. So either we can perform oxidation reaction, in which we use different acids like performic acid. Okay, different acids are used and we oxidize. So we have a SS bond, as you can see here, we have a SS bond, and after oxidation it will become OS. There will be a OS bond. Okay, so there will be a breaking of disulfide bond and we'll have a OS molecules. Okay, so this disulfide bonds will break into by oxidation, and we can also do reduction reaction, and this reduction reaction is performed by DTT or mercaptoethanol. Okay, where this disulfide angle will be reduced, and after reduction we'll get SH. Okay, we'll get SH sulfahydryl. Okay, by oxidation we'll get OS, and by reduction we'll get sulfahydryl groups. Okay, now this is the first step, that we have to break all the disulfide bonds in the protein, because they form the major core of uh, the 3D three-dimensional structure of the protein. Then after breaking the disulfide bond, the second step is we will break down, we'll cleave the protein in short fragments. Okay, we'll cleave the protein in short fragments, and this fragmentation can be done by different enzymes. Okay, different cleaving enzymes like trypsin. Okay, as you can see here, they have used different they have used trypsin, and they have got uh, fragment 1, 2, 3 and 4. So these are the, so trypsin is a very special enzyme, and it will cleave after lysine and arginine. After every lysine and arginine residues, it will cleave. Similarly, we can also cleave with different enzymes like chymotrypsin or cyanogen bromide, as mentioned here. So cyanogen bromide also cleaves after methionine residue. Okay, so and these so after cleaving with, so here we have used two cleaving enzymes, trypsin and cyanogen bromide. So with trypsin we are getting 4 fragments, and with cyanogen bromide we are getting 3 fragments, 1, 2 and 3. And as you can see, these fragments have overlapping sequences, that is T1 and T2 is overlapping with C1, whereas T4 is overlapping with C3, and T3 is overlapping with C2. Okay, see, so these are the overlapping sequence which will help us to backtrack the protein sequence. Okay, the whole protein sequence. Now we are left with different fragments. Now we are left with these different fragments. So we can analyze these different fragments by using two methods. And the first method here is either we can use Edmond degradation or Sanger's method. So both these methods analyze proteins from N-terminal end. Okay, all these proteins are sequenced or analyzed from the N-terminal end. So in the Edmond degradation we use dinitrofluorobenzene. Okay, dinitrofluorobenzene. So the structure is also very simple. We have a benzene, we have a fluorine, and we have a nitro group. Okay, NO2, 2NO2 group. Okay, and this dinitrofluorobenzene will react with the amino acid with the amino end. So this is our amino acid. Let's say this is the N-terminal, first amino acid in the N-terminal. Okay, so it will react and it will form a complex. This DNFB will form a complex with this amino acid, and this complex is a colored complex. Okay, it's a yellow colored complex which can be either measured by HPLC or TLC. Okay, we can measure this by this complex by HPLC or TLC. Okay, um, then similarly, yes, okay. Similarly, in the Sanger's method uses. Okay, sorry. So this is the Sanger's method. This is the Sanger's method, and this is okay. So in Edmond method, we use um, phenyl isothiocyanate, also known as PITC. So this PITC is also one of the reagent. Um, it will react with the amino acid. Okay, it will react with the amino acid, and again it will form a complex with the amino acid. Okay, and so this is our amino acid sequence. This PITC will also form a complex with the amino acid. So we have this N-1 sequence now, because the PITC will form a complex with the N-terminal amino acid. Similarly, we can also analyze this same way via either HPLC or TLC. Okay, apart from the N-terminal sequencing, we also have C-terminal sequencing also. And this C-terminal sequencing can be done by using carboxypeptidase. So this is the enzyme which will cleave amino acid residues from the C-terminal. Okay, it will cleave amino acid residue from the C-terminal end. Okay, it will cleave amino acid residues from the C-terminal end, and then each amino acid is released, which could be either analyzed by HPLC or TLC again. Okay, so this is how the sequence we can analyze different what is the protein sequence. Now the secondary structure of so the secondary structure of protein, that is the alpha helix and beta sheets, can be analyzed by CD spectroscopy. So CD spectroscopy is nothing but so we have different chiral amino acid present in the protein structure, and these chirality will help us with the with the help of the chirality present, the circular dichroism or so this CD uses the so this CD utilizes this chirality present in the amino acid, different amino acid except glycine. Glycine doesn't have any chiral carbon, and it will give us spectra. So the CD is generally measured in 190 to 250 nanometer, 190 to 250 nanometer wavelength. Okay, so this is the wavelength which is used for studying the secondary structures, and we'll get specific peaks for different secondary structures. For example, for alpha helix, as you can see here, so this is the alpha helix peak. So for the alpha helix, so this is alpha helix, red one. It has two negative peak. Okay, this one peak is okay. It has two negative peak, and the intensity with a very small intensity, and this negative peak are at 222 nanometer and 208 nanometer. Okay, these are the two less intensity peaks, negative peaks, and when we have one high intensity peak at 192 nanometer. Okay, this at 192 nanometer we have a high intensity peak. So this peak is due to pi transition. Similarly, Um, this 222 peak is due to N to pi transition. So if you have a peptide bond, so this N to pi is generally for the peptide bond. Okay, peptide bond in each structure. So the alpha helix will have two negative peaks and one high positive peak. Similarly, for beta sheet we have one negative band and one positive band. So this is the band blue. So the negative band is between 216 and 218. Okay, so the negative band is here. This is the negative. So for this is the beta sheet. Blue is the beta sheet. So the negative band is between 216 and 218. This is one negative band, and we have one positive band at 195 nanometer. Okay, this is at 195 nanometer. And then we, apart from alpha helix and beta sheets, we have um, other structures like which are random ordered or random coiled structures. Okay, they are not completely alpha helix or completely beta sheet, they are just random coiled structures. Okay, and these random coiled structures are also captured in the CD spectra, and the CD spectra for the those random coil structures are, we get here. This green color is for the random coil. Okay, so we have one positive peak here at 218 nanometer, and we have a negative peak at 200 nanometer. Around 200 nanometer we have a negative peak. So this is how you can characterize different secondary structures. For alpha helix you will get a characteristic peak like this. So in a protein there will be mixture of both alpha helix, beta sheets and random coil. So accordingly you will get a mixture of CD spectra, and not entirely it will match to this CD spectra. So this CD spectra is specifically for alpha helix, beta sheets and random coil separately, but for a protein where you have mixture of all these secondary structures, the spectra will be little more complex. So the next spectroscopy technique in order to analyze the secondary structures is infrared spectroscopy. So in infrared spectroscopy we generally measure the molecular stretching and the molecular vibration. So IR is generally used to measure molecular stretching and vibrations. Okay, so here are the different peaks which we can get in IR spectroscopy. So for alpha helix we have peaks between 1657 and 1648 wavelength. Similarly for beta sheet we get peak at 1641 to 1623. Similarly, unordered is the random coil which we get, and the peaks are scattered between 1657 and 1642. So this is how the peaks for the infrared spectroscopy will look like. So we'll have two amide peaks, and these two amide peaks these are the characteristic amide peaks which will be seen for the protein structures. Okay, for alpha sheets as well as beta sheets. So here are two peaks here. So and these two peaks are merged. So these are the merged peaks, and the max for them is 1655 and 1634. These two peaks are merged together for the amide 1 peak, whereas for amide 2 the peak is around 1500. So the tertiary structure can be analyzed using either X-ray crystallography or NMR. So one of the important characteristic of X-rays is that the wavelength is much lower, and it will give you much more atomic detail as compared to the light or electron microscopy. As you can see here, the light microscope which we have, it has the wavelength of 300 nanometer, which we use to visualize different cells, subcellular organelles. Okay, whereas electron has wavelength around 10 nanometer. So with electron microscope we can visualize cellular architecture, like subcellular uh, uh, architecture of different proteins, organelles. um, Whereas X-ray has very small wavelength. Okay, and this will help us to analyze or visualize different atomic details of protein. So so the main steps in X-ray crystallography is, first we need to have a purified protein, and with this purified protein we'll do the step of crystallization. Okay, so the crystallization step is the most important step, where you generate different crystals. Okay, and these crystals can be generated in lab by two methods. One method is hanging drop method, and the second method is sitting drop method. Okay, so these are the two different methods in which you can generate crystals of different proteins, and this is a major crucial step. After getting nice crystals, you can do X-ray crystallography, and the basic principle is that the X-ray, the crystals, the electrons in the crystals in the nuclei will diffract the X-rays, and it will give you a pattern. Okay, it will give you a different pattern depending upon your molecule, and then we collect the data. Now the next important step is structure solving. So based upon the whatever data or diffracted pattern which we have got, we need to solve the structure of the protein. Okay, so we have different methods to solve the structures, different uh, softwares which we can use to solve the structures, or people also do it manually. Then further we can build a model and then refine the model by validating it with the available data, or we can use Ramchandran plot to validate. Okay, so this is the basis of X-ray crystallography. Then the tertiary structure analysis can also be done using NMR spectroscopy. So the NMR spectroscopy utilizes uh, it utilizes the set distances of the atomic nuclei. Okay, so X-ray crystallography utilizes the electrons in the atom. Okay, how the X-rays are diffracted because of electrons, whereas the NMR spectroscopy, it utilizes the set distances from the atomic nuclei. This is very important and differentiating factor from X-ray crystallography to NMR spectroscopy. So so these are the different steps in NMR spectroscopy, that we need pure protein in order to do NMR spectroscopy. Otherwise, you get different peaks for impurities. And so So we get so from NMR crystallography, from NMR spectroscopy. So then finally we need to dissolve this pure protein. For NMR spectroscopy, we'll dissolve this pure protein into solvent, and then we'll do NMR spectroscopy and data collection. So we'll get different peaks. Okay, so we'll get different peaks for different types of tertiary structures, okay, present in the, it will give us all the atomic molecular level peaks present, okay, in the tertiary structure of the protein. Okay, then the next step is sequential resonance assignment and diffraction data collection. So the machine will give again do the diffraction data collection depending upon the distances of the atomic nuclei, and will get a chemical shift of the different secondary structures as shown here. Now the third step is so the first step is okay, so after doing this, the first step is uh, from NMR spectroscopy we'll get conformation, geometric conformation constraint. Okay, geometric conformation constraints, and these constraints could be either bond distances, angles, or the orientation of the bond. Okay, depending upon all these factors we'll get these different peaks, okay, of the particular tertiary or the secondary structure of the protein. Then uh, after getting that, we need to estimate the structure by using different programs or analyzing these chemical shift. Okay, so we need to assign this chemical shift based upon different programs. So we will have a standard structure which will have a standard peaks. Then we'll assign those peaks to our data, okay, our molecule. And after this, after assigning, they should we should also consider that they satisfy all the dihedral angles. Okay, they must satisfy dihedral angles and all the bond lengths. Okay, so it is very important that they satisfy the bond lengths and dihedral angles. Okay, so after getting after solving the structure and getting assigning all the chemical shift, we need to satisfy the bond lengths and the dihedral angle. We can also validate it by using Ramchandran plot again. So now what is the difference between so sorry. So X-ray crystallography and NMR has different. So these both methods have different pros and cons. So the first properties of X-ray, that will get a solid, will get a static structure. Okay, we'll get a static structure, that is um, the way it has crystallized, we'll get a static image of the crystallized protein. uh, okay, whereas in NMR the we'll get a dynamic solution structure. Okay, we'll get dynamic solution structure because our protein is dissolved in a solvent, that is why we'll get a dynamic solution structure. Okay, the next point is X-ray. The major cons in X-ray crystallography is that we need to have a crystal with us. okay, and it is very difficult to get a protein crystal. Okay, so if we don't have a crystal, we cannot do X-ray crystallography. Whereas for NMR, we don't have any requirement of do not require any crystal structure. Okay, we need to just have a pure protein, uh, which without any impurities, which can be directly analyzed using um NMR spec. The third important point is, for in X-ray crystallography we get very high resolution of our structure. Okay, the resolution is very high, whereas in NMR we get average resolution. Okay, so these are the pros and cons for the each of the technique um, in order to find out the second tertiary structures by X-ray and NMR spectroscopy. So the quaternary structure here, so this so as I mentioned earlier, quaternary structures is formed by different subunits which are attached together. Okay, so we have a primary structures, which is amino acid joined together by peptide bond. Okay, then we'll get a secondary structure in which we have alpha helixes or beta pleated sheets. In the tertiary structure, we have both alpha helixes and beta sheets joined together. Okay, and in the tertiary structure, we have different subunits of these tertiary structures together, forming a quaternary structure. Okay, and these could be n number of subunits. So in order to determine these quaternary structures, we can use simple technique like SDS page. Okay, so SDS page is a denaturing page, where the denaturing agent here is sodium dodecyl sulfate. It's a surfactant. So what this surfactant will do, this surfactant will denature this protein. So if we have a quaternary protein like this, suppose we have 4-unit quaternary protein, uh, in presence of SDS and heat, we need to heat it in order to get denatured. This quaternary structure will get denatured into different subunits. Okay, it will denature into different subunits, and now this subunits can be analyzed by either SDS page, or we can do different chromatographic technique also. But with the SDS page, what we'll get different bands. Okay, with SDS page we'll get different bands if these subunits are of different molecular weight, or we can get only one band, okay, if these are of same molecular weight. So let's say if these two protein 1 and 2 are of same molecular weight, whereas this A and B are of different molecular weight. So 1 and 2 will form a bond here because they are of same molecular weight, whereas A and B will form two different bands, okay, as shown here. So these are the different ways in order to um, find out whether the protein has quaternary structure or not. And this graph here shows the elution time or elution volume. So as I mentioned, after denaturing we can run this in different chromatographic in column chromatography, and we can collect different elutes, and this different elutions will get depending upon either which type of chromatographic technique which if we are using. Okay, and we'll get different elutions. So so here we are getting 6 elutions, which mean there are 6 different subunits present. Okay, and these peaks denote each subunit, and as the eluent is having that subunit, there is increase in absorption, which suggests that the elution contains different subunits. Okay, then the next is we can also analyze the protein based upon so these all were the experimental methods, which the in previous slide we studied all the experimental methods in which we can analyze proteins. So we studied um, X-ray crystallography, NMR spectroscopy, circular dichroism, and infrared spectroscopy. So these were all experimental methods wherein you have to purify the protein, we have to do different spectroscopy technique, do crystallization. There are other ways also you can estimate the protein structures in where the using molecular modeling molecular modeling approaches, wherein to get experimentally to wherein the uh, so when the experiments are not possible or the protein uh, we cannot experimentally find out or purify the protein, we can use molecular modeling approaches where we can predict that where we can predict the structures of different proteins. Okay, so uh, the molecular modeling approach can be is of two types. One is ab initio method, ab initio molecular modeling method, and the next is homology modeling approach. Okay, so in the homology modeling, first we'll begin with homology modeling. It utilizes homologous structures which are already present in the database. So homologous structures which are already present in the databases, it utilizes those structures. Okay, um, and it uses the similarities in those structures. It uses similarities in those structures and use those similarities as a template in order to form the base data. Okay, it uses similarities to form base data template. Okay, so this is the general structure of homology modeling, that we should have the knowledge of homologous structures which are present in the database, and from that database we must find out what are similarities present in the with our structure and the already available structure in the dataset, and then we can use that available dataset as a template in order to analyze the protein, the X unknown protein. Okay, for example, if I have a mutant protein, Y, let's say Y, or we can say mutant protein X. So this mutant protein has some mutation, and there is some conformation change in the there is some change in the structure of protein. So this change can be analyzed by checking the homologous structure, that is the wild type structure present in the database. Okay, then we can map the similarities in the database, and from that wild type protein structure we can form a template, and then similarly we can predict what our X protein will fold, or what conformation or how stable our X protein would be. Okay, so this is the basis of homology modeling, whereas in ab initio modeling, molecular modeling, we have no homology. If our protein of interest is completely new, if the protein is completely new and we have no knowledge about the protein, then we use this ab initio molecular modeling. Okay, so it will predict and it will predict a structure, and we can verify that structure based upon energy parameters, different energy parameters, or we can also predict using Ramachandran plot whether they are satisfying the dihedral angles and the bond length. And also we can do dynamic simulations, dynamic folding and unfolding simulation in order to dynamic folding and unfolding simulation in order to find out how this model is working. So then we can understand finally that or predict the structure or folding of this new protein or new found protein, which for which we don't have the database available. um, yeah, so these are the steps used in homology modeling. So we have we need to first find the query sequence. We can use different tools like NCBI databases, and there are different databases available for different proteins. So NCBI is one of the most used database. Then we'll select the template. Okay, we'll select the template, and then we can align the template. Okay, there are different tools again in order to align also the template. So suppose we have this wild type sequence, then we can align this with our mutant sequence, and after aligning we can build a model, and then we can validate the model by using different tools. Okay, so this is the general structure of molecular modeling, homology modeling. So any questions? Any questions? Do you have any questions? Okay, then we'll begin with the assignments. Last previous year assignment question. So this is the first question. The alpha helices and beta sheets are connected in tertiary structure of a protein through A. disulfide bond, B. unstructured loop, C. hydrogen bonds, and D. hydrophobic interaction. So anybody knows what is the answer for this? You can unmute yourself and answer. It's pretty simple. Yes. So I'll explain you. So these are the tertiary structures. So tertiary structure is formed by alpha helix and beta sheets. Okay, and these alpha helixes. So this is alpha, one of the alpha helix, and this is beta sheets. Okay, and these together are connected via some unstructured loops which are shown here. Okay, these are the unstructured loop. Similarly, this is one more beta sheet, and it is connected to this alpha sheet via this random structure. Okay, this is random structure. Okay, it will just give us the shape of the protein. It will just tell us, it will help us forming the shape or the structure of the protein. So the correct answer is B, unstructured loops. Okay, so they will just orient these sheets or helices in a particular direction. Okay, they could be either turns, beta turns, or random structures which are present in just orienting these secondary structures like beta helix and alpha sheets. Alpha helix and beta sheets. Okay, so the correct answer is unstructured loops. The type of bond used in joining amino acid in the protein is peptide bond. Very simple. Okay. So we have the two amino acids here. This is one amino acid and this is the second amino acid. Okay, these together are formed by hydrolysis. There is a hydrolysis of water and we have a peptide bond, which is a amide bond, CONH bond. So the correct answer is peptide bond. The part of amino acid that varies among the 20 amino acid is alpha carbon, amine group, carboxyl group, side chain or R group. So we have, so again very simple. So we have the structure of amino acid as we have carboxyl, we have NH2 amino group, we have R group, and then we have hydrogen here. So this R group, which is also known as side chain, it will vary for different amino acids. So R group could be either hydrogen, it could be methyl group, it could be phenylalanine group. Okay, the R group could be anything. Okay, so the correct answer is D, that among all the 20 amino acids, the side chain or the R group varies. The next question is the amino acid that contains a thiol group is. So thiol group is nothing but SH group, and the SH group is only present in one amino acid, that is cysteine. Okay, SH group, this is the thiol group. Okay, which is present in cysteine. So correct answer is cysteine. For serine and threonine, we have hydroxyl group, and proline is the cyclic amino acid. Okay, the R chain is is connected to the amino group of the amino acid. So the cysteine has thiol group. The Ramachandran plot helps in determining protein primary structure, identifying amino acid chains, defining allowed rotational angles for amino acid in protein structures, studying protein folding. So Ramachandran plot again, it will define the different dihedral rotational angles, which are phi and psi angles. Okay, so again psi are C and C alpha angles, whereas phi is N and C alpha angle. Okay, and so these are the different angles. So it will define, by defining I mean that it will have fixed angles, like the left-handed alpha helix will have fixed angles as shown here. Okay, similarly, these rotational angles are fixed for different secondary structures, and uh, so different protein structures, they always satisfy this different psi and phi angles. Okay, so the correct answer is C, defining allowed rotational angles for amino acids in protein structures. Next, match the reagent with the respective cleavage point and choose the correct option. So again very simple. So we have hydroxylamine. Okay, so hydroxylamine is the one which is tricky. We know that cyanogenamide will cleave after methionine. Okay, so cyanogen bromide will cleave after methionine. Trypsin will cleave after lysine and arginine. Okay, so trypsin will cleave after lysine and arginine. So 2,3C. Okay, and pepsin will cleave after leucine, phenylalanine, and tyrosine and arginine. Okay, so pepsin will cleave after A4A. So the correct answer is hydroxylamine will cleave after asparagine. Okay, the correct answer is B. Okay, you have to remember different cleavage points for different enzymes. So PITC is again used in Edmond degradation. Okay, PITC reacts with the terminal amino group and to give, so we'll it gives, so it will react with this is the PITC. It will react with the N terminal amino group, right? It will react with the amino group and we'll get phenyl carbomyl derivative. Okay, this is phenyl carbomyl derivative. So the correct answer is D. Okay, so we'll have a phenyl carbomyl derivative here. Finally, this derivative will get hydrolyzed. Okay, it will get hydrolyzed and the peptide will be released and we'll have this PTH. Okay, we'll have this PTH and this compound will give yellow color. So they will estimate this in order after Edmond degradation. The role of turns in protein structure is forming alpha helixes, uh, altering the direction of the polypeptide chain, serving as the primary structure of protein, forming beta sheets. So forming alpha helixes and beta sheets is not possible. The turns are not possible for forming helices or beta sheets. Uh, Serving as the primary structure, no, primary structure is solely formed by amino acids and peptide bond formation. Altering the direction of the polypeptide chain, this is correct, as I mentioned earlier also. So because of this turn, so suppose let's consider this helix. uh, this is how it is moving. Okay, and because of this turn now, we have a beta sheet at this, we have beta sheet at this direction instead of going in any other direction. Okay, so these turns are doing nothing but they are altering the direction of the polypeptide chain, of this polypeptide chain. So now this beta sheet will go in this direction. Okay, they will form in this direction and not in any other directions. Okay, so they are just altering the direction of the polypeptide chain. Different protein achieve similar 3D conformations despite variation in amino acid preferences in turn regions by altering the primary structure, forming alpha helices predominantly, favoring beta sheet formation, adjusting the length of amino acid in the turn. Again, different proteins achieve similar 3D structures despite of varying because so here they are asking that we have different protein and achieve similar 3D conformation despite the variation in amino acid preferences in turn region by, so again we have, so all this, the 3D conformation uh, will will be depending upon the turns, how can we adjust the length of the amino acid in the turn region. Okay, so similarly again the same principle again here, that so if the turn is very steep, okay, if the turn is very steep, then we'll have longer elongated proteins, whereas if the turn is spreaded out, as you can see here, the angle of turn here is huge. Okay, as compared to this turn. Okay, as compared to this turn, the angle of turn is huge. That is why we are getting uh, a different shape of protein here. We are getting a square shape of protein. This is a top view of a protein, whereas if we observe from here, we will get a filament like structure of protein. Okay, this is the filament like structure of protein. Okay, so so the same thing again, adjusting the length of amino acid in each turn. The appearance of two overlapping bands in the amide 1 region at 1634 and 1655 in infrared spectrum of polypeptide indicates. So yeah, so this band here. Okay, so these are the two overlapping bands here. Okay, these bands here indicate Yeah, so these bands here indicate the presence of both alpha helix and beta sheet structure. Okay, it indicates the presence of both alpha helix and beta sheet structures. Okay, so these bands comprises of both alpha helix and beta sheets. The vibrations that primarily contributes to amide 1 band in the IR spectrum of the protein. So this is because of carbonyl stretching vibration. Okay, so the amide 1 bond is found by carbonyl stretching vibration. The free R factor in a protein structure determination is utilized as, sorry, mm, huh. So sorry. Yeah, the free R factor in a protein structure determination is utilized as it assesses the agreement between calculated and observed data. Okay, this is in X-ray crystallography that so we'll have what is during crystal solving we'll have calculated and observed data. So this R factor, free R factor, it is used in order to determine this observed and calculated data during solving of crystals. Different types of atomic nuclei recognized in NMR spectroscopy is by their chemical shifts. Okay, is by their chemical shift of and coupling constants. Okay, so based upon the distance, set distances from the nuclei, there will be different chemical shifts and coupling constants depending upon the different structures. Okay, so it is analyzed, this is recognized by NMR spectroscopy. The critical factor for the reliability of homology model is, so one of the major requirement for the homology model as we studied is the degree of sequence similarities. Okay, we need to have homologous sequences. We need to have homologous sequences between the target. Let's say this was our X mutant protein and the template proteins. So these template proteins which we got from the database. Okay, we need to have some sequence similarity in order to we need to have sequence similarity or the homologous sequences in order to have the to do homology modeling. The construction of 3D structure in the homology modeling is well guided by sequence alignment, molecular dynamic simulation, Ramachandran plots, and loop modeling. So the correct answer is sequence alignment. Okay, so this is the end of the assignment questions. Um, okay, so any questions? Okay, then I'll stop the meeting. Thank you for attending.